Hello, welcome to Biology, the scientific study of life. Ascomycetes, mycologists have described more than 32,000 species of Ascomycetes, fungi, and the phylum Ascomycota from a variety of marine, freshwater, and terrestrial habitats. The defining feature of Ascomycetes is the production of sexual spores in sac-like asci. They are commonly called sac fungi. Unlike zygomycetes, most Ascomycetes bear their sexual stages in fruiting bodies or ascocarps, with range in size from microscopic to macroscopic. The spot-forming asci are found in the ascocarps. Ascomycetes vary in size and complexity from unicellular yeasts to elaborate cup fungi and morels. They include some of the most devastating plant pathogens. However, many Ascomycetes are important subgroup, particularly in plant material. More than 40% of all Ascomycetes species live with green algae or cyanobacteria in symbiotic associations called lichens. Some Ascomycetes from mycorrhiza with plants, still others live between Mesophylla cells in leaves, where they release toxic compounds that apparently help protect the plant from insects. One of the best studied Ascomycetes is Neurospora crassa, a bread mold. Though Neurospora grows readily on bread in the wild, and it's found growing on burned vegetation. Biologists in the 1930s years used Neurospora to formulate the one gene, one enzyme hypothesis. To them, this Ascomycete continues to serve as a model organism. In 2023, its entire genome was published with 10,000 genes. The genome of this tiny fungi is 3/4 the size of the Drosophila genome and 1/3 the size of the human genome. However, the Neurospora genome is relatively compact, having few of the stretches of non-coding DNA that occupy much space in the genomes of the humans and many other eukaryotes. Biologists have found evidence suggesting that Neurospora has a genomic defense system that prevents junk DNA from accumulating to harmful levels. Although the life cycles of various Ascomycetes group differ in the details of their reproductive structures and processes, there are some common elements. Ascomycetes who reproduce asexually by producing enormous numbers of asexual spores called conidia. Conidia are not formed inside sporangia, as are the asexual spores of most zygomycetes. Rather, they are produced externally at the tips of specialized hyphae called conidiophore. Often is in clusters or long chains from which they may be dispersed by the wind. Conidia may also be involved in sexual reproduction, fusing with hyphae from a mycelium of a different mating type. As occurs in Neurospora, fusion of two different mating types is followed by plasmogamy, resulting in a heterokaryotic, non-septated bulge or ascogenium. Also, synoiotic ascogonium extends hyphae that are partitioned by speta or septa, forming dikaryotic cells, each with two haploid nuclei representing the two parents. The cells at the tips of these dikaryotic hyphae develop into asci within an ascus. Karyogamy combines with two parental genomes and then meiosis forms four genetically different nuclei. This is followed by a mitotic division forming 8 ascospores in many asci. The 8 ascospores are lined up and a row in the order in which they formed from a single zygote nucleus. The ascospores develop in and are eventually discharged from the ascocarp. In contrast, the life cycle of zygomycetes, the extended dikaryotic stage of ascomycetes, and also Basidiomycetes provides increased opportunity for genetic recombination. For example, in some ascomycetes, certain dikaryotic cells fuse repeatedly, recombining genomes and resulting in a multitude of genetically different offspring from one mating event. Notice that. Et voilà. This is the life cycle of Neurospora crassa in ascomycetes. Notice that. Fungi have a powerful impact on ecosystems and human welfare. Decomposers, fungi are well adapted as decomposers of organic material, including the cellulose and lignin of plant cell walls. However, almost any carbon-containing substrate, including jet fuel and oil paint, can be consumed by at least some fungi. Fungi and bacteria are primarily responsible for keeping ecosystems stocked with the inorganic nutrients essential for plant growth. Without these decomposers, carbon, nitrogen, and other elements would become tied up in organic matter. Plants and animals that eat them could not exist because elements taken from the soil would not be returned. Symbionts. Fungi form symbiotic relationships with the plants, algae, cyanobacteria, and animals. All of these relationships have profound ecological effects. Mycorrhiza are enormously important in natural ecosystems and agriculture. Almost all vascular plants have mycorrhiza and rely on their fungi partners for essential nutrients. It is relatively easy to demonstrate the significance of mycorrhiza by comparing the growth of plants with and without mycorrhiza. Foresters commonly inoculate vine seedlings with ectomycorrhiza fungi to promote vigorous growth in the trees. Fungi, animals, symbiosis. Some fungi share their digestive services with animals, helping break down plants' material in the guts of cattle and other grazing mammals. Many species of ants and termites take advantage of the digestive power of fungi by raising them in farms. The insects scour tropical forests in search of leaves, which they carry back to their nests and feed on the fungi. Bon. The fungi break down the leaves into a substance the insects can digest in some tropical forests. Notice this figure. Et voilà. In some tropical forests, the fungi have helped these insects become the major consumers of leaves. The evolution of such farmers' insects and that of their fungi crops have been tightly linked for well over 50 million years. The fungi have become so dependent on their caretakers that in many cases they can no longer survive without the insects. Lichens are symbiotic associations of millions of photosynthetic microorganisms held in a mass of fungi hyphae. They form a surface-hugging carpet that can be found growing on rocks, rotting logs, trees, and roofs in various shrub-like, leaf-like, or in clustering forms. The photosynthetic partners are typically unicellular or filamentous green algae or cyanobacteria. The fungi component is most often an ascomycete, but several Basidiomycete lichens are known. The fungi usually gives a lichen its overall shape and structure, and tissues formed by hyphae account for most of the lichen's mass. The algae or cyanobacteria occupy an inner layer below the lichen surface. The merger of fungi and algae or cyanobacteria is so complete that lichens are actually given scientific names as though they were single organisms. More than 30 or 13,500 species have been described, accounting for a fifth of all known fungi. In most lichens that have been studied, each partner provides something the other could not obtain on its own. The algae provide carbon compounds. The cyanobacteria also fix nitrogen, and to provide organic nitrogen, the fungi provide their photosynthetic partners with a suitable environment for growth. The physical arrangement of hyphae allows for gas exchange, protects the photosynthetic partner, and retains water and minerals, most of which are absorbed either from airborne dust or from rain. The fungi also secrete acids, which aid in the uptake of minerals. Fungi pigments help shade the algae or cyanobacteria from intense sunlight. Some fungi compounds are toxic and prevent lichens from being eaten by animals. The fungi of many lichens reproduce sexually by forming ascocarpus or basidiosexual or basidiocarpus. Lichens algae reproduce independently of the fungi by asexual cell division. As might be expected of diel organisms, asexual reproduction as a symbiotic unit also occurs commonly either by fragmentation of the parental lichens or by the formation of psoridia, small clusters of hyphae with embedded algae. Phylogenetic studies of lichens DNA have helped illuminate the evolution of this symbiosis. Molecular studies published in 21 support the hypothesis that all living lichens can be traced to three original associations involving a fungi and a photosynthetic symbiont. The same studies also indicate that numerous free-living fungi descended from lichens forming ancestor Bansalium.